Great. Thank you very much. I appreciate the opportunity. So Aardvark can you see my screen, everybody? Okay, great. Aardvark is a San Diego-based company. We're developing ARD-101, which is a bitter taste receptor agonist. Bitter taste receptors are found throughout your body, highly expressed in the gut. And we believe our drug is working through gut-brain signaling via induction of endogenous secretion of CCK. And just almost recapitulating what Dr. Feiner had stipulated before, we think that there's a difference between appetite and hunger. Appetite is the pleasure-seeking drive for eating, and hunger is a pain avoidance. We believe they're regulated separately. GLP-1s, we believe, are more anti-appetite, and our drug via gut-brain signaling through CCK, which projects vagal afferents to the nucleus of the solitary tract in the brainstem, modulate hunger, which we believe is more relevant empirically for Prader-Willi syndrome. One of the reasons why we had conviction that this is a good avenue to pursue is because there's evidence that deficient endogenous CCK signaling is evident in Prader-Willi subjects. Dr. Tony Holland, probably 32 years ago, elucidated that the CCK level of Prader-Willi individuals are similar to baseline individuals. However, upon eating food, that rise is not proportional as in a normal individual, and they're missing the feedback loop to induce satiety, which is what CCK normally does. We believe our drug is a secondary pathway to induce localized endogenous CCK production, which then facilitates the induction of satiety. There's other lines of evidence that the other sequelae for Prader-Willi is related to deficient CCK signaling. I'm not explaining everything, but at least it's consistent with the other clinical features. CCK is related to gut motility, gallbladder secretion, and also preservation of lean body mass. And as we know, Prader-Willi individuals have extremely delayed gut transit time. They have gallstones at an early age. They also have higher body fat percentage, even at a given BMI. And also, gut-brain CCK is related to an anti-anxiety phenomenon as well too. Folks with partial vagotomies actually have increased anxiousness as well. We believe maybe related to deficient afferent CCK signaling. So that's why we think mechanistically we are optimistic that ARD-101 could address the multiple symptoms. So CCK has been tried as a therapeutic strategy before, but the limiting feature has been on-target off-tissue toxicity because CCK receptors are found throughout the body, and neurotoxicity and pancreatitis was evident in other approaches using systemic CCK analogs. Our drug induces CCK receptor activity in the lumen of the gut. It induces basolateral site secretion, which is localized. And because of the anatomic secretion of CCK there, we believe we have a more selective approach, which lends itself to the safety. Another feature of the drug that's actually very unique is that the drug is largely gut restricted. So 99% of the drug is retained in the gut as an oral drug. And that's actually not an issue. It's actually the target of our drug is in the lumen of the gut. So by doing that, it actually lends itself to the safety. And because it stays in the gut largely, you don't have to worry as much theoretically about drug-drug interactions or liver or renal clearance. We've had over 70 patients, including normal healthy volunteers, obesity patients, post-bariatric surgery patients, as well as Prader-Willi individuals in our clinical trials. And thus far, we've only had very limited AEs, only 3 grade 2s, which are the urticaria alleviated by antihistamines. No renal or hepatic safety limitations. We believe the therapeutic index of our drug is over 80. We've had good results in the early phase 2A, an open label single arm study in Prader-Willi individuals at 2 clinical sites. Of the 12 patients in that low dose, and this is at the low dose of 200 milligrams BID, we saw 11 out of 12 respond with reduced HQ-CT of average of 7.1 points. 4 participants had a near complete resolution of their hyperphagia. We also saw trends of reduced body fat percentage and clinical reports of reduced anxiousness and other clinical features that are encouraging. And we also do believe that higher doses will invoke a higher response, given the fact that our DC50 knowledge of our drugs indicates to us that higher doses should do better. So as we go forward, we're going to initiate a pivotal study of 90 patients, and we're going to advance to 800 milligrams BID as our intended dose. I know I'm short on time, so I want to keep to the 6 minutes. So We saw a reduced ghrelin, and this is a statistically significant result of ghrelin. This is in general obesity patients. This is not Prader-Willi, but we saw reductions of ghrelin, which is what is opposite of what you'd expect in the GLP-1 drug. So we measured the two different ways through fasted states versus placebo, and also at 28 days versus screening. Here it is showing our preclinical data suggesting that higher doses should do better in our review, so that we're very encouraged by the higher dose. So anyway, so we have a pivotal study that we're planning, and we intend to initiate this very soon, a multinational pivotal trial, which hopefully will enable us to file an NDA for this. And I know I'm out of time, so that's it. I think I'm under 6 minutes.